Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. On this occasion, allow me to introduce myself. My name is Anita Oktari from School of Analysis Presentasi Bandung, Indonesia. I represent colleagues in my research team that are Haidir Safrullah from Health Science Schools of Dharma Husada Bandung, Indonesia, and Nizar Hairumi from School of Analysis Presentasi Bandung, Indonesia. Our research is about the use of BETALAIN dyes, beetroot extract, beta vulgaris linnaeus as alternative dyes SAFRANIN substitute in bacteria simple stain. Here are the background of our research. A simple coloring can show the arrangement bacteria such as chains or streptococcus, grape-like or staphylococcus, cuboid or sarcina, and rod shape. The dyes commonly used are methylene blue, gentian violet, carbolfuchsin, and safranin. Waluyo 2010. In modern times, the use of natural dyes is very popular. One of the natural dyes that can be used as a natural dye to replace safranin is BETALAIN, contained in red beetroot or beta vulgaris linnaeus, Vulgaris 2000. In a previous study conducted by Ibrahim 2015, extracting BETALAIN dye from beetroot extract as staining for malaria thin blood smears, and it was found that a 10% concentration of beetroot extract could be used as staining on malaria thin blood smears. In the preliminary test, researchers used beetroot extract with a concentration of 50%, which had been concentrated with a Oktari vacuum evaporator. The result is that the extract can stain bacteria and the color of the bacteria is clearly visible. Here are the method of our experiment. There are 3 steps. From one step, the process beetroot drying using an oven at 50 degrees Celsius for 48 hours. And the second step is the production of beetroot extract, beta vulgaris linnaeus by ethanol solvent 1 to 3. The filter produced from 3 times of extraction is put together, checked for pH, then concentrated using a rotary vacuum evaporator at 60 degrees Celsius until the solvent stops dripping from the condenser. And the third process is the making 0.25% safranin dyes, bacterial preparation, and simple coloring with safranin dyes. As a treatment in this study, staining bacteria with extract stain, the concentration used were 5%, 7.5%, 10%, and 12.5%. Here are the results and discussion from the research. Figure 1, A for Salmonella typhi with 5% beet extract staining. This is the picture. And B, Staphylococcus epidermidis with 5% beet extract staining. And figure 2, this is Salmonella typhi with 7.5% beet extract staining. And the figure B, Staphylococcus epidermidis with 7.5% beet extract staining. In this picture, the bacteria are still not clearly visible. For Salmonella typhi, these bacteria are rod-shaped. And for Staphylococcus epidermidis, these bacteria are focus Staphylococcus. The next result from this experiment, that is figure 3, Salmonella typhi with 10% beet extract staining from picture A, and the picture B, Staphylococcus epidermidis with 10% beet extract staining. And figure 4 from A, Salmonella typhi with 12.5% beet extract staining, and figure B, Staphylococcus epidermidis with 12.5% beet extract staining. In this picture, the bacteria are starting to be seen clearly. Okay, the next result from this experiment. Figure 5. This is Salmonella typhi with safranin staining, and 5A, and the 5B, Staphylococcus epidermidis with safranin staining. In this picture, serve as a control in the treatment of this study. So we can compare the result from figure 1, 2, 3, 4, and 5. Here are the discussion of our research. In this study, the beetroot extract was alkaline, and the safranin, which was the control, was also alkaline. They were both alkaline, so they could bind to acidic microorganism cells. With such treatment, the transparent microorganism cell would lose color. During the staining process, the positive charge on the basic dye will be related to the negative charge on the cell, so the microorganisms are more clearly visible. Mastuti, 2010. The final concentration of safranin as a control in simple staining was 0.25%. But in this study, the beet extracts used started from a concentration of 5%. Because if the beet extract was diluted to a concentration of 0.25%, it could not stain Salmonella typhi and Staphylococcus epidermidis. Simple staining using an alternative solution of beetroot extract with a concentration of 10% and 12.5% obtained almost the same result as the control, namely 0.25% safranin. The color of the bacterial cells was clearly visible and red, almost the same as the color produced by 0.25% safranin. Then, staining using an alternative solution of 7.5% beetroot extract also produced a color that was already clearly visible in red, but not as concentration as the control with 0.25% safranin solution. Meanwhile, staining using an alternative solution of 5% beet extract obtaining different color result with 0.25% safranin control. So, the conclusion of our research is, based on the result of research and data analyzed, it can be concluded that betaline dye of beetroot extract or beta vulgaris lineus can stain Salmonella typhi and Staphylococcus epidermidis for simple bacterial staining. The concentration of beetroot extract that produce optimal color contrast and is the same as the control, namely 0.25% safranin solution for coloring Salmonella typhi and 10% extract for Staphylococcus epidermidis. This is the end of our presentation. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.